Today we'll be talking about mutations. But first, let's take a look at what results from transcription and translation in our original DNA strand. Here we have our DNA strand written in the 3' to 5' direction. In order to reach our mRNA strand, we'll have to first identify the reading frame. We can do this by scanning across from left to right and then right to left to find our start codon, AUG, which in a DNA strand will be ATG. We also know that our amino acid sequence is 4 amino acids long, so we can double check that we have the right reading frame through this as well. So, while scanning from right to left, I can see that we have an ATG here followed by 3 other codon sequences and a stop codon sequence, TAA. Let's see what they end up coding. I flipped the original coding strand to read to the 5' to 3' direction, and now we can write the corresponding mRNA sequence. Using our coding dictionary, we can see that our amino acid sequence is as shown. Okay, now let's dive into some different types of mutations. The first one we'll look at is called a silent mutation. In this mutation, although there is a change in our nucleotide bases, our amino acid sequence stays the same. For example, we can see here that there is a change in our original DNA strand from C to T, which is shown as U in the mRNA. This change from C to T is called a transition because it's a change from one type of base, in this case a pyrimidine, to another nucleotide that is also a pyrimidine. And we can see here that there is no change in our original amino acid sequence because GUU still codes for valine. This is sort of like driving and taking a different route to reach the same location. A missense mutation is when a change in an amino acid sequence results in the coding of a different amino acid. For example, here, our G has turned into a C. This is called a transversion because our base changed from a purine to a pyrimidine. And we can see that our amino acid sequence has changed from lysine to asparagine. This is as if you took a wrong turn while driving and were unable to reach your final destination. Another type of mutation is called a nonsense mutation, where there's a stop in translation before intended. For example, here we can see that there's a change from A to T in our DNA strand, which resulted in a U in our mRNA strand. This would be a transversion, and the new UAG codon is a stop codon, which would halt translation before our actual stop codon is supposed to. This is sort of like if you decided to go for a drive and just stopped before reaching your destination. Finally, the last type of mutation we'll be looking at today is called a read-through mutation, where a stop codon is changed into another codon. In this example, our A changes into a C, which is a transversion. UAC codes for tyrosine, and our stop codon is deleted, which means that our translation will continue. In our driving analogy, this would be like if you decided to just skip going to the grocery store and instead decided to drive past it to the beach. And that's all the mutations we'll be going through today. As you can see, a lot of these mutations result in significant changes in our amino acid sequence, which will most likely cause defected or mutated proteins that will be unable to properly operate. Even a small point mutation can have a significant effect on our bodies.